Okay, so welcome back to this next video in which we are discussing depression and antidepressants. Okay, so we're in the process of trying to discuss what is known about the cause of depression. Okay, now I've said that the most powerful lead for the cause of depression is most likely the overfunction in the anterior cingulate cortex. Okay, we're now talking about the hypothalamic pituitary adrenal axis. Okay, now another thing that has been noticed in a lot of people who suffer from depressive episodes is that the hypothalamic pituitary adrenal axis is overactive. Now, why is it overactive? Well, we know exactly what seems to go wrong, okay, and to explain what goes wrong, I need to tell you a little bit more about the physiology of the hypothalamic pituitary adrenal axis, okay, specifically the feedback mechanisms. So, we have seen that in the fear response, what happens is an original stressful or fearful stimulus is processed by the brain. The brain sends this information in the processed form to the amygdala, and the amygdala says, shall we initiate a fear response to this or not? Okay, if it decides that this stimulus that it's been sent information about is worthy of a fear response, it initiates the fear response, and one of the things it does in this is it activates the paraventricular nuclei in the hypothalamus. Okay, the paraventricular nuclei then release corticotropin releasing hormone into the capillaries that supply them with blood, and these capillaries then reconverge into a hypothalamic hypophyseal portal vein, okay, which takes the blood down to the anterior pituitary, and then it opens back up into capillaries, and then the corticotropin releasing hormone can leave the capillaries and stimulate cells in the anterior pituitary. And the specific type of cell that's going to be stimulated by corticotropin releasing hormone are known as corticotrophic cells. Okay, these will be stimulated to release the next hormone in this pathway, which is adrenocorticotropic hormone, ACTH. Okay, this will then go back into these capillaries and then will go in a vein into the systemic circulation. The ACTH will then make its way down to the cells of the adrenal gland, okay, specifically the cells in the zona fasciculata, which is the second layer of the adrenal cortex, and it will stimulate the cells in this second layer of the adrenal cortex to release the hormone cortisol. Cortisol, we have then discussed, reduces inflammation, raises blood glucose, and also raises your energy level and also your cognition. It sharpens your cognition so that you can think more clearly. Okay, right, which are all beneficial for fight or flight situations. Okay, however, there is a negative feedback loop which I didn't mention before, which is that cortisol now goes back up both to the paraventricular nuclei, okay, and also it goes up to the corticotrophic cells. Okay, and it stops them from releasing their hormones. Okay, so cortisol will go up to the paraventricular nuclei here, okay, and it will stop them from releasing corticotropin releasing hormone. Okay, so you're going to stop releasing corticotropin releasing hormone. It will also go directly to the trophic cells rather, and stop them from releasing adrenocorticotropic hormone. Okay, and through both of those mechanisms, you're going to stop the activation of the release of more cortisol. So cortisol stops its own release, basically. And this is a nice negative feedback loop. This stops cortisol levels in the blood going too high, basically. This is a negative feedback loop. In a lot of people suffering from a depressed period, okay, you find that this negative feedback loop is broken. Okay, so cortisol in the blood no longer goes to the paraventricular nuclei and no longer goes to the corticotrophic cells, and it no longer stops them from releasing their hormones. Okay, so there is no negative feedback response anymore, and therefore these cells will continue to release their hormones and you'll continue to trigger more cortisol release. Okay, and what is that going to result in? It's going to result in cortisol going far too high, basically, for a long period of time. Okay, so a lot of people with depression end up with chronically high cortisol levels. Okay, now how do we know it's specifically this portion of the hypothalamic pituitary adrenal axis that is broken? Well, basically you can put in an incredibly powerful synthetic glucocorticoid known as dexamethasone, okay, which does the same thing as cortisol, but it's synthetic rather than being made in the body, okay? And it will, well, in a healthy person, dexamethasone will cause cortisol levels within the blood to collapse, basically, okay, because it would go up to the paraventricular nuclei and the corticotrophic cells of the anterior pituitary, and it would stop them from releasing their hormones, which would then stop the triggering of the release of cortisol by the zona fasciculata cells. Okay, so cortisol levels within the blood would go down upon administration with dexamethasone. That does not happen in these depressed patients, so we know that it's some sort of problem with the negative feedback loop, basically. Okay, right, so in depression then, there seems to be this other lead that cortisol levels are too high. Now, this draws parallels between what is happening in depression and what happens in chronic stress, okay? So if you are chronically stressed, what's going to happen is you're going to be continually activating your hypothalamic pituitary adrenal axis, and this also leads to increased cortisol levels. So this too high cortisol level is very similar to what happens in people who are just chronically stressed, and there is a link between chronic stress and also depression. Now, we don't know whether there is any sort of causal relationship between these two, or whether it's just chance, basically, whether depression truly is a problem with the anterior cingulate cortex, which then somehow through unknown mechanisms leads to this problem with the HPA axis, and this HPA axis problem then is just going to contribute to the symptoms of depression, giving similar symptoms to chronic stress, or whether there's a more interesting story here about how chronic stress is related to depression. Okay, we don't know at the moment. At the moment I'm telling you all that we know, which is that in a lot of people who are depressed, there is this problem with the hypothalamic pituitary adrenal axis. What has caused this, we don't know. Whether this is involved at all with the anterior cingulate cortex, we don't know. Whether chronic stress is the cause, we don't know. Okay, so what I'm going to tell you now is what is the effect of having chronically high cortisol levels and how this might contribute to some of the symptoms of depression. Okay, particularly the problems with memory. Okay, right. So we come to the hippocampus then. Basically, cortisol causes, well, it's believed to cause what's known as hippocampal atrophy. Okay, sorry, not hippocampal atrophy, atrophy. Okay, atrophy is sort of like, well, I don't know what it is. Hippocampal atrophy, it's a word similar to atropine, I suppose. Hippocampal atrophy. Atrophy means the wasting away of something. Muscle atrophy means the wasting away of muscle, basically. So hippocampal atrophy means that the hippocampus gets smaller. Okay, now this is a common finding that in people who are depressed, you find smaller hippocampi, okay? And it's believed that potentially the reason for this is that the chronically high cortisol levels cause the hippocampal atrophy. They cause the shrinking of the hippocampus. Okay, so what I'm now going to describe to you is the anatomy of the hippocampus in more detail than we did it previously. Okay, previously I just described it to you as this sausage-shaped thing that was inside the temporal lobe and wasn't visible from the outside. Now I'm going to go into that in more detail. Okay, so I'm going to do a coronal section again. In fact, I'm going to draw basically the same picture as we have here, but I'm going to forget about this bit up here and just draw the bit down here now. Okay, and go into this bit down here in more detail, which is where the hippocampus is going to be here. Okay, so we're looking at a coronal section then of the temporal lobe. Okay, so here, let's say this is the lateral fissure here, and here is the cerebral cortex of the temporal lobe coming here. So let me just make sure that everyone is familiar with what I'm drawing here. I'll draw a picture of the brain from the side. We are taking a little cut down here. We're doing a coronal cut. I'm putting my knife like this, okay, and I'm cutting through the brain, and then I'm looking at what we see when we pull these two portions apart and look at the inside. Okay, and I'm looking at this portion here. So at the moment, this portion that's highlighted in green is this portion here. This is the portion of the cerebral cortex that lines the inside of the lateral fissure here. So there's another piece of cerebral cortex here, and then you've got the parietal lobe above. Okay, so if I colour this in pink, like so. Okay, right, so this is the lateral fissure here. Or rather, I'm pointing at the moment to the piece of cerebral cortex there. This gap here, this is the lateral fissure. Okay, right, so what's going to happen is the cerebral cortex is going to come round like so, and then down here you're going to have something known as the collateral sulcus. Okay, so you've got a little invagination of the cerebral cortex inwards here. Okay, then you have a piece of cerebral cortex known as the parahippocampal gyrus here. Okay, and it folds up like so, and I'll label all of these bits up later, and then it's going to fold round, and this is where I said that the hippocampus really is just the edge of the cerebral cortex. So now we're getting to the edge of the cerebral cortex, and it's going to fold round into the hippocampus. So here is the beginning of the hippocampus. It continues round, it folds all the way around, and this is still basically just cerebral cortex. Okay, and it folds round, it forms a clump of cells like so. Okay, and then you've also got another clump of cells here. Okay, like so, and like that. Okay, and these are the key structures that are going to be part of the hippocampus. This portion here isn't strictly speaking part of the hippocampus. It's part of what's known as the hippocampal formation, but I'll define those terms later on. Okay, so let's colour bits in then. So, this portion here in blue, which is still very much so just part of the cerebral cortex. Okay, this portion is known as the parahippocampal gyrus. Okay, because it's parallel to the hippocampus. And I should stress that this is running all the way along in the internal aspect of the temporal lobe here. Okay, so it's not just one little section, it goes on and on all the way down here. Okay, so you've got this all like so. Okay, right, so this is the parahippocampal gyrus here in blue. Okay, so I'll underline that in blue. Then I'll actually label up this little sulcus that we've got, this little invagination of the cerebral cortex here. This is the collateral sulcus here. Okay, now, after the parahippocampal gyrus here, the first portion that you've got here, which I'll colour in purple, this is known as the subiculum. Okay, so this here, this is the subiculum. Okay, and again, this isn't part of the hippocampus yet. This is part of a broader thing called the hippocampal formation. Now, often when people say the hippocampus, they really mean the hippocampal formation, but I'll go into that difficulty in words in a moment, once I've labelled up all of the different bits here. Okay, right. Then, after the subiculum here, what you've got is the CA1 portion, okay, and all the portions that are labelled up CA, these are the portions that are actually part of the hippocampus. I can't remember what CA actually stands for. It stands for something like conus accumbens or something like that, which means ram's horn in probably Latin, okay. And that's because the hippocampus originally, when they found it, they thought it looked like a ram's horn when you look at a coronal section like so. Okay, so that's where the CA comes from, but for our purposes, just know that the CA, anything that is labelled CA something, is part of the hippocampus, basically. Okay, so here's the first bit, CA1. Okay, then following CA1, you've then got CA2 here. Okay, in orange. Okay, then you've got a little section known as CA3, which I'll again put in vivid purple so that it shows up. Okay, so this is also part of the hippocampus, so all the portions that are labelled CA, those are the proper portions of the hippocampus. Okay, and then this little bit here in green, that's CA4. Okay, like so. And that's the end of the proper portions of the hippocampus, so CA1 to 4, strictly speaking, that is what we mean. Well, that is what we should mean when we say hippocampus. Okay? Some people, well, often when people say hippocampus, they don't just mean CA1 to CA4, and sometimes to stress that you literally just mean CA1 to CA4, people will put the hippocampus and then they'll put proper afterwards, so they'll call CA1 to CA4 the hippocampus proper, the proper hippocampus, okay? And that's because even though we shouldn't, when people say hippocampus, generally they don't just mean CA1 to CA4, they mean CA1 to CA4 along with the subiculum, along with this other portion that I haven't named here yet. Okay, so this final portion that surrounds CA4 here, which is now coloured in yellow, this is known as the dentate gyrus. Okay, so this is called the dentate gyrus. Okay, right. So now onto this difficulty with regards to words. Okay, basically the hippocampus, strictly speaking, means CA1 to CA2, CA3, and CA4, and that's it. The proper term, which means the hippocampus along with the subiculum, along with the dentate gyrus, is the hippocampal formation. Okay, so the hippocampal formation means CA1 to CA4 plus the dentate gyrus, okay, and then also plus the subiculum. Okay, so that's the strict word that people should use. However, people generally just use the word hippocampus to mean the hippocampal formation, which is why some people use hippocampus proper to mean the CA1 through CA4, and then just use hippocampus to mean the subiculum, CA1 to CA4 plus the dentate gyrus. Okay, so that's a problem with words that you should be aware of. Okay, so often when I say the hippocampus, what I really mean is the hippocampal formation, and that's not just me, that's everyone, okay? So be aware that the terminology here is a little bit icky. Okay, right, so that's the hippocampus, and it is extraordinarily involved in memory, particularly spatial memory, as I talked about earlier, so the ability to remember how three-dimensional space is stuck together, basically, and to recall that and be able to remember your way back to the car where you've parked it in some unfamiliar city. Okay, right, so what does high cortisol level do then to the hippocampus? Well, basically it causes the hippocampus to get smaller. Okay, so cortisol being too high for a very long period of time starts to cause the hippocampus to become smaller. And I've said that the word for this, the fancy terminology for this, is hippocampus atrophy. Okay, so the hippocampus atrophies. Right, now there are two theories as to how too high cortisol levels causes hippocampus atrophy. Okay, one of them is that cortisol being very high literally just kills the neurons. Okay, so the first theory is that cortisol kills the neurons of the hippocampus. Okay, and that's how you get hippocampus atrophy. Okay, the other theory is that it doesn't kill the neurons, instead it stops neurogenesis. Okay, now the kill neuron theory is very simple to understand, so I'm not going to explain that any further. The stops neurogenesis theory is a little bit more difficult to understand, so this one I'm going to discuss in a little bit more detail. Right, so if you had learnt neuroscience 20 years ago or something like that, there would have been absolutely no concept of this word neurogenesis. 20 years ago, the neurons that are in your brain were the same neuron, well, it was believed that the neurons in your brain throughout your entire life are the same neurons as you had when you were born. Okay, so the cells are not replaced and they do not divide. Okay, there is absolutely no cell division in the brain, was the central dogma on which neuroscience sat. Okay, now, however, we are aware that in certain portions of the brain, brain cells, neurons, are actually replaced. And one of those places where you actually have brain cells being replaced is the dentate gyrus of the hippocampal formation. Okay, so again, here we are viewing the dentate gyrus as being part of the hippocampus. Strictly speaking, it's part of the hippocampal formation, but we view it as being part of the hippocampus. Okay, so basically in the dentate gyrus, certain cells are being replaced on a regular basis, okay? And neurogenesis means the creation of new neurons. Okay, right. So I'm going to tell you a little bit more about the structure of the dentate gyrus so that we can understand which of the neurons are being replaced continuously in the dentate gyrus. So if we draw this out a little bit more, okay, the dentate gyrus looks like this, okay, and it has 3 separate layers. So I'm drawing this yellow structure out in more detail, but now what we're going to acknowledge is that it's not all one layer. Instead, you have multiple separate layers, okay, like so. Okay, this is the portion where CA4 is. So here is CA4 here. I'll put this in green like so. So this is CA4. Okay, now the outermost layer, the layer that is furthest away from CA4, the layer that's pointing down towards the subiculum and CA1, this is known as the molecular layer of the dentate gyrus. Okay, so this is the molecular layer. Underneath the molecular layer, this middle layer here, is the granule cell layer. Okay, and often if you see these on images, they will abbreviate them down to ml for molecular layer. The next layer is the granule cell layer. Okay, that will be abbreviated down to GCL. Okay, and then the layer underneath the granule cell layer is the subgranular zone. Okay, and the subgranular zone will usually be abbreviated down to the SGZ. Okay, so let me bring this up. So subgranular zone down to SGZ. So let's colour in these separate layers. Okay, so we'll have the subgranular zone in orange here. Okay, we'll have the granule cell layer here in red. Okay, like so. And then we'll have the molecular layer here in vivid purple. Okay, so basically in the granule cell layer, as the name suggests, you are going to have granule cells. Okay, it is these cells which are being replaced in the hippocampus on a regular basis. Okay, so granule cells are being replaced in the hippocampus on a regular basis. The idea then is that if cortisol stops the production of new granule cells, okay, so if you no longer produce new granule cells, then the old granule cells would eventually die, okay, but now they would be replaced by nothing. Okay, and that would also cause the wasting away of the hippocampus. So it's not that you're killing the existing neurons, it's that you're stopping the production of new neurons. That would also explain how the volume of the hippocampus overall goes down. Okay, whichever theory is correct, what seems to be the case is that chronically high cortisol levels is causing hippocampal atrophy, okay, which then is responsible for a reduced ability to remember things, so memory dysfunction. Okay, so memory goes down, and that could explain why memory is dysfunctional in people with depression. Okay, right, so that now concludes that discussion of the dysfunction in the hypothalamic pituitary adrenal axis and the effect that it has on the hippocampus. Okay, right, we are now going to move on to another theory of depression. Okay, so we've talked about the anterior cingulate cortex. Okay, we've now talked about this dysfunctional hypothalamic pituitary adrenal axis. They don't fit together at the moment, but they're not, they're not contradictory. They could be working together. Okay, potentially the dysfunctional anterior cingulate cortex could cause the dysfunction in the hypothalamic pituitary adrenal axis. Okay, we're now going to see a totally different hypothesis, okay, which is the monoamine hypothesis, which is a beautiful hypothesis. It's extremely simple, okay, but it's largely starting to be abandoned. Okay, it's almost certainly oversimple. When you see it, you will think, good gracious, this is, you know, too simple, basically. Okay, however, it's a very useful model because it allows us to understand completely how the antidepressants work and why they work. Okay, so pharmacologically it's an incredibly successful model. Okay, it's just being left behind by the functional neuroimaging studies and things like that. Okay, right. So, before I can explain to you the monoamine hypothesis of depression, I firstly need to explain the monoamine systems within the brain. Okay, so I'll start off by describing the 3 different monoamines in the brain, and then I'll describe how they are released into the brain and how they regulate arousal. Okay, so the monoamine systems then. So, the 3 monoamines, okay, and what this name means is that the molecules, which we will see later, we'll see their molecular structure, they contain a single amine group. Mono means one, amine means an amine group. So monoamine, they contain a single amine group. Okay, so the 3 monoamines that are within the brain, okay, are firstly serotonin. Okay, that's a very important one, particularly in depression. Okay, now serotonin also has another name. It's also known as 5-hydroxytryptamine. Okay, and we'll see how that name makes sense when we see the production and the structure of it later on. Okay, for short, 5-hydroxytryptamine is also abbreviated to 5-HT. Okay, right. The other monoamine neurotransmitters are dopamine, okay, and also noradrenaline. Okay, now noradrenaline is the old name for this neurotransmitter. It's got a new name as well, which is commonly used, which is norepinephrine, but they're the same thing. Okay, right. We will see the structures of these 3 molecules later on, but for now, just know their names. Okay? What I want to explain is what these 3 neurotransmitters are doing within the brain, because they are not used in the classical way that neurotransmitters are used. Okay, so I'm firstly going to describe the classical way that neurotransmitters are used, okay, which is the way that most neurotransmission within the brain works. And then I'm going to describe what these neurotransmitters are doing and how they differ. Okay, right. So, let me get another piece of paper. Right, okay, so classical neurotransmitters then. Okay, the classical idea of a neurotransmitter is that you have some axon terminal here, okay, like so, and this is the presynaptic neuron here, okay, and this presynaptic neuron will release the neurotransmitter, okay, which I'll abbreviate as NT, when an action potential arrives. So the action potential will arrive down the axon, it will arrive in the axon terminal, and the action potential arriving here will trigger the release of neurotransmitter. The neurotransmitter will then cross the synaptic cleft to a postsynaptic structure here. So this is the postsynaptic structure, which will most likely be a dendritic spine on a dendrite of a neuron. Okay, so just to make sure everyone knows what a dendritic spine is. Okay, basically, neurons have these structures known as dendrites. Okay, so if I draw a picture of a neuron, the typical sort of neuron looks like this. It has these processes coming off the cell body like so, known as dendrites. Okay, then it has a large process coming off the cell body known as the axon. Okay, so here is the axon, and here is the axon terminal right at the end of the axon here. Okay, here's the cell body with the nucleus here. So basically, the dendrites collect information from other neurons, okay, and they integrate this information together to decide whether this neuron needs to fire an action potential down its axon. Okay, now, the dendrites are covered in little processes that look like this, which are known as dendritic spines, and these are the structures which other neurons synapse onto and cause little effects on, okay? So this is how information from loads of other neurons converges on this single neuron here, and this single neuron will integrate all of this information together to decide whether it's going to fire an action potential down its axon. Okay, right. So the classical idea then of a neurotransmitter is that the neurotransmitter will now act on receptors on the postsynaptic cell, okay, and these receptors will generally be ligand-gated ion channels, okay, and when the neurotransmitter binds to the ligand-gated ion channel, which I'll draw as a little cylinder here, it will cause that ion channel to open, and either the ions that will be conducted through the ion channel will be sodium ions, okay, and therefore they will have a positive charge, or they will be chloride anions and they will have a negative charge. Okay, now the main two neurotransmitters in the brain that work like this are glutamate, which is the main excitatory neurotransmitter of the brain. It makes it more likely that neurons are going to fire action potentials. Okay, glutamate is released by presynaptic neurons onto postsynaptic neurons, will act on glutamate receptors, which will open and allow sodium into the cell. Okay, positively charged ions in the cell then makes it more likely that the neuron will fire an action potential. Okay, so that's the main excitatory neurotransmitter of the brain, and then the main inhibitory neurotransmitter of the brain is GABA. Okay, now GABA will bind to GABA receptors on the postsynaptic neuron, and when GABA binds to those receptors, they will open and they will allow in chloride anions. So glutamate is going to allow in sodium cations, and GABA is going to allow in chloride anions, and negatively charged ions will make it less likely that this neuron is going to fire an action potential, and therefore will have an inhibitory effect. Okay, so the classical sort of idea is that loads of neurons will synapse onto this neuron here, and that they'll all be releasing either a stimulatory neurotransmitter or an inhibitory neurotransmitter, and then it will be receiving sodium and chloride anions all over the place, and it will have to integrate all of this information to decide what the net result is. Has it received positive charge or negative charge? And if it receives enough positive charge overall that hasn't been cancelled out by the negative charge, it will then fire an action potential. So that's the sort of classical view of neurotransmitters, and these are the two classical neurotransmitters within the brain, glutamate and GABA. They do work like that. Okay, the monoamines, serotonin, dopamine, and noradrenaline do not work like that. Okay, they do not work on ligand-gated ion channels, at least not majorly, in the way that glutamate and GABA do. Okay, instead, the idea of the monoamine systems in the brain is that you have specific little loci, okay, located in the brainstem, which contain neurons which are capable of releasing the monoamines. So that's actually the first thing that I should say, that very few neurons in the brain actually do release the monoamines. Most of them release either glutamate or GABA. Okay, there are very few neurons in the brain which actually do release serotonin, dopamine, or noradrenaline. Okay, and they're located in specific collections, okay, specific nuclei which are located in the brainstem. Okay, the idea then is that these neurons send their axons all over the brain, and they release the monoamine neurotransmitter all over the brain, and the monoamine neurotransmitter doesn't have an effect just on one neuron. Okay, it has an effect on loads of neurons in the local area, and it changes the function of the entire brain, basically. Okay, so these monoamines are what are known as regulators, and they regulate things like arousal. So they're regulators of things like arousal, so how vigilant you are, how awake you are, and hence they are really modulating the function of the entire brain rather than just modulating the function of a single neuron. Okay, so that's how they're different to these neurotransmitters. These neurotransmitters in this classical sort of setup are just changing the function of one neuron. These are being released onto not one neuron but loads of neurons all at the same time. They don't have specific synaptic contacts like this. Instead, what you have is just the axon terminal like this, which will just not have a postsynaptic neuron, and it just releases the neurotransmitter into the extracellular fluid, and this can then go and act on all sorts of different neurons, basically loads of them. Okay, and it can modulate the functions of much more than just one neuron. Okay, so really these monoamine systems are modulating the arousal states of the entire brain, basically. They're modulating the function of the entire brain. Okay, right. So hopefully this will become more clear when I show you the specific nuclei that are involved in releasing serotonin, dopamine, and noradrenaline. So let's start with serotonin. Okay, serotonin unfortunately is the most difficult because there are loads of nuclei which can release serotonin, and we're not going to go through each one individually. There are two main groups of nuclei that release serotonin, and those I am going to show you. Okay, so again, if we imagine having a sagittal picture of the brain, so again what I'm imagining doing, if I get my old picture of it, I'm going to take this sort of section again, a sagittal section down the great longitudinal fissure. Okay, I'm going to draw another one of these pictures to show the nuclei which release serotonin. Okay, right, so let's do this here. I'm not going to do it quite as fancy as that other one. I'm not going to show as many things on it. Okay, so here is our right cerebral hemisphere coming round like so, and then down like here. Okay, then we'll have the temporal lobe sort of sticking out here. Okay, then we'll have the pons here. Okay, like so. And then we'll have the medulla below like so. And then the brainstem down here. Okay, and then we'll have the cerebellum sat there. Okay, right. So there are two main groups of nuclei which are involved in secreting serotonin. One is located right at the top of the pons up here, almost in the midbrain region, okay, which will be above there. And then one is located right at the bottom, near where the medulla is, and sort of bordering into the upper medulla. Okay, right. So these ones, which are closer to the nose, these are known as the rostral raphe nuclei. Okay, and the ones that are closer to the tail are then called the caudal raphe nuclei. Now I want to stress again, there are far more raphe nuclei than just two. Okay, so all of the nuclei which secrete serotonin are known collectively as the raphe nuclei. There are absolutely loads of the things, but they can be grouped into these two separate groups. The rostral raphe nuclei, which I'm drawing as one blob, but in reality, you know, there's loads of the things. Those are located up here at the top of the pons, and then the caudal raphe nuclei, which are located right down here at the base of the pons going into the medulla below. Okay, right. Now, neurons in these raphe nuclei produce serotonin, and they are the only neurons in the brain which do produce serotonin. None of the others use it. Okay? And what they do is they send their axons all over the place. So, for instance, the caudal raphe nuclei, they send axons into the cerebellum, okay, behind us here. So remember this structure here, which we haven't really talked that much about because it's not really too important for us in depression. This is the cerebellum, okay, here, okay, and I'll colour in that fibre in purple as well. So you're getting fibres going into the cerebellum. You also get fibres going down the brainstem and down into the spinal cord, okay. So these neurons are sending fibres into all these sort of places, and they're going to be releasing serotonin into all sorts of places, all down the cerebellum and all down the lower brainstem and spinal cord, and they're going to have modulatory functions on a huge number of different neurons in those lower portions of the central nervous system. Okay, the rostral raphe nuclei, they send fibres into the cerebellum as well, okay, and they also send fibres up into the cerebral hemispheres, which generally go round like so. Okay, so you'll have loads of these fibres. I'm only showing one, but you know, you'll have absolutely loads of the things. Okay, following these same sort of pathways and delivering serotonin to different portions of the brain. So the idea really is that you've got serotonin in the extracellular fluid surrounding neurons, and this has a basal role on their activity. okay, in regulating their activity. Okay, now these systems then control that level of serotonin depending on your situation, and therefore they can regulate the entire brain's function. Okay, so this is the structure then for serotonin. Okay, let's now discuss the corresponding systems for dopamine and noradrenaline. So I want to show you again where the nuclei which release dopamine and noradrenaline are as well. So we'll do noradrenaline next. Okay, noradrenaline, the two main nuclei which secrete noradrenaline are in the pons, okay, and they are the left and the right locus ceruleus. Okay, so what we're now going to do is we're going to take a cross-section of the pons. So if you imagine cutting down like this, okay, and another piece of anatomy terminology, when you take a section like that, it's known as a transverse section. So we've seen coronal sections now, we've seen sagittal sections, and now we're seeing transverse sections. So we're going to take a transverse section of the pons like so. We're going to imagine cutting through in a plane that is sort of transversely like that, and having a look at the pons underneath. Now, something that I haven't really exaggerated in any of my pictures so far is that off the back of the pons you have major structures coming off, okay? So let me draw the pons from the side again, just before I do the transverse section. Okay, really the pons has these very large structures coming off from the back of it, like so. Okay, and it will have another one on the other side. So here's the one on the other side. So let me try and make this one thicker. Okay, and then here's the one behind. Okay, like so. Okay, so you have these large structures coming out from the back of the pons, like so. So this is the pons here. Okay, so this is the pons, and these structures are transmitting fibers into the cerebellum here. Okay, so these are what are known as the middle cerebellar peduncles. Okay, and you have the left middle cerebellar peduncle and the right middle cerebellar peduncle. So cerebella. So this is the middle cerebellar peduncle. Right. Okay, so when we take a transverse section through like this, we are going to see these middle cerebellar peduncles. So that's all that I'm mentioning these middle cerebellar peduncles to you for, so that you understand when I show you the transverse section what these projections coming out the back are. Okay, so the pons then, when you take a transverse section of it, it looks kind of like this. So this is the front of the pons, so this is the portion towards the eyes. Okay, and then to the back of the pons you then have the cerebellar peduncles coming out here. So here's the left cerebellar peduncle and here's the right middle cerebellar peduncle. Okay, and there are superior and inferior cerebellar peduncles as well that are further and up and down as well, respectively. Okay, right. Now, the portions where these noradrenaline producing neurons are contained are far back in the pons, okay, shown here. So these portions that I've shown in blue here, these are the two portions which contain neurons which produce noradrenaline, okay, and these are known as the locus ceruleus. Okay, or at least one of them is known as the locus ceruleus. I don't actually know what the plural is, so this is the right locus ceruleus, and this is the left locus ceruleus. I would imagine loci cerulei or whatever, okay, but that sounds really weird. So this is the left locus ceruleus, and this is the right locus ceruleus. Okay, so these have noradrenaline containing neurons in them, and they send axons all around the brain, and again, this noradrenaline has a basal regulatory function, and these locus ceruleuses are controlling how high the basal concentration of noradrenaline is in the extracellular fluid of the brain. Okay, right. Finally, let's now talk about where the nuclei which secrete dopamine are. Okay, so the nuclei which secrete dopamine are contained within the midbrain. So again, let's go back to our cross-section of the midbrain. Okay, so we've drawn this picture before. So this is the Mickey Mouse picture again. So here are the superior colliculi at the back here. Here are the cerebral peduncles right at the front, these processes that extend out like so. Okay, and then it'll fold around like so. So you've got the left side of the midbrain and the right side of the midbrain. Then we know in the middle we've got the periaqueductal grey surrounding the cerebral aqueduct here. Okay, so let's colour in the periaqueductal grey and red. So now what we want to show is the nuclei which secrete dopamine, and in fact I'll just put on the red nuclei first. Okay, so those are the landmarks that we mentioned previously. So here are the red nuclei in turquoise. Okay, right, so the key dopamine producing neurons are in two nuclei that are on either side, here and here, and these are known as the substantia nigras. Okay, so we've got a left substantia nigra and a right substantia nigra here. Okay, so this is the substantia nigra. So that is Latin for black substance. Nigra means black, substantia means substance. Okay, so this shows up very, very dark when you see it, and this is real. If you look at the brain, you can tell where the substantia nigra is, because it really does look very, very dark. Okay, and these neurons are dopamine producing neurons in the substantia nigra. Now there is another collection of dopamine producing neurons which sits in the middle here, in between the two red nuclei. Okay, and this is the ventral tegmental area. Okay, here. So both of these areas, the two substantia nigras, and then also this one in the middle here, which is called the ventral tegmental area. Both of these areas contain dopamine producing neurons and are sending their axons out all over the brain and controlling the basal concentration of dopamine. Now, the ventral tegmental area then is often abbreviated as the VTA. Okay, right, so those are the monoamine systems of the brain. Okay, right, so now I'm ready to tell you the monoamine hypothesis. Okay, and I'm not ready yet to tell you exactly where the monoamine hypothesis came from. That will come later, once we've studied the serotonin and noradrenergic neurons in more detail. Okay, but at the moment I can tell you the monoamine hypothesis. The monoamine hypothesis was that depression was caused by too low levels of the monoamines in the brain. Too low levels of serotonin, too low levels of noradrenaline, and maybe too low levels of dopamine. The main two are serotonin and noradrenaline. The monoamine hypothesis is that depression is caused by too low levels of serotonin and also too low levels of noradrenaline, maybe too low levels of dopamine as well. And I will give you what sparked that theory later on. Okay, in the next video what we will do is we're now going to discuss the neurons which release serotonin and noradrenaline in more detail. We're going to abandon dopamine. Dopamine isn't too involved in depression. It is part of the monoamine hypothesis for reasons that I'll explain later on. Okay, but all of the antidepressants seem to work on serotonin and noradrenaline, so we'll abandon dopamine. But we are now going to study the serotonin releasing neurons and the noradrenaline releasing neurons. This will lead us to a better understanding of where the monoamine hypothesis came from, okay, and then we'll discuss all of the antidepressant drugs which actually all work by increasing serotonin and noradrenaline in the brain, which is a major supporting argument in favor of the monoamine hypothesis. Okay, even though the monoamine hypothesis is losing favor as we do more and more studies and find that really are monoamines actually lower in people with depression, and the answer's kind of no. Okay, but still, what we know with regards to how the antidepressants work, it seems to be that they raise serotonin and noradrenaline.